Alright guys, this one hopefully will be a lot shorter than the normal one, not 15 minutes, because today we're only going to finish up going over the cytoskeleton, and that would be our microfilaments and our intermediate filaments. Yesterday we talked about the microtubules. Wow, I say yesterday. Okay, you're not really there. So here we go. Microfilaments are the thin solid rods, but they're about 5 to 7 nanometers in diameter. So these are the smallest of parts of the cytoskeleton. Their structure is two intertwined strands of actin protein. So they're tightly wound and it's like a tensile rod. And I'll show you a picture of this. Okay, so we're right now here in the middle. And if we look at microfilaments, we also call them actin filaments because they are composed of this protein called actin. And we're going to talk a lot about that. Some of you, especially if you've taken anatomy and physiology, should remember the term actin. Okay, again, you can see in comparison, their diameter is much smaller than that of a microtubule. And this is what it looks like, this actin subunit. Not very large, okay, the two intertwined, so it's coiled up like a tensile rope there. And we can see this is the dyed image of all the microtubules in a cell. I'm sorry, I just said microtubules, microfilaments. So the microfilaments here you can see on this TEM are within the microvilli. Microvilli are found in your small intestine to help increase surface area for absorption of nutrients during digestion. So we can see here that our microfilaments, again actin filaments, compose a lot of the cytoskeleton for each one of those. And again, we can see our microfilaments, they're stained green in this section. Okay, so they compose quite a bit of our cell. So what are its functions, that tiny little thin rod? The first function is muscle contraction. For those of you who were in anatomy and physiology, hopefully you remember the words actin and myosin, and that the attachment and the sliding of these proteins is what causes our muscles to be able to shorten, and then when they relax, to go back to their normal state. Okay, so we have muscle contraction with the action of myosin. They also allow for cytoplasmic streaming in plant cells. If you've ever looked, I'm trying to recall if you guys have done that with a plant cell, where you looked at maybe maybe some algae before, or Elodea, when you looked at and you could see the chloroplast moving through the cell, you're going to see some cytoplasmic streaming. And that's the movement of the cytoplasm around the cell. The other function is the pseudopodia. Pseudo means false, podia means foot. For example, this is a movement in amoeba, and I'm going to show you a picture of this in just a second. Then we have the cleavage furrow, which is the formation for cell division. This is occurring in animal cells when we form that. You might remember that a cell starts like this and then begins to split and pop. Okay, where it begins to pinch, that is where we get our cleavage furrow. And then finally, maintenance and changes in cell shape. So again, we're in a very broad area where it functions as, again, that's why we call it the cytoskeleton. Cyto means cell, and then skeleton of the cell, because it's going to provide help with the shape. So let's take a look at this pseudopodia. We can see here, this is my amoeba. In the inner cytoplasm, the soul here, which is the liquid-like portion, and that would be more of the gelatin area, we can see the movement of actin subunits, which causes it to extend. So it's like that crawling, that kind of motion there. And then in cytoplasmic streaming in cells, again, here, where we can see the movement of those microfilaments causing the cytoplasm to move around the cell, and therefore you would see chloroplast moving as well. The last part of the cytoskeleton that we're going to look at are the intermediate filaments. And these guys are called intermediate filaments because they're right in between microfilaments and microtubules when it comes to size. So they are fibrous proteins that are supercoiled into thicker cables and filaments, and they're 8 to 12 nanometers in diameter. They're made from several different types of protein. And again, so let's take a look at them on our table. The proteins are supercoiled, so we are slightly larger than the microfilament, but not as large as a microtubule. Actually, and when we say several different proteins, one of those proteins is that they're all part of the keratin family, and you might have heard of keratin before because it's the protein in your nails. It tends to last a very long time. It doesn't degrade very quickly, very tough, very resistant, very sturdy. And this sturdiness allows for the following. Okay, the main function of these guys, they're specialized for bearing tension, just like a microfilament. And so again, they provide help with maintenance of cell shape, and they hold organelles in place. Okay, so it's that intermediate filament that helps to hold them in place so they're not going all over berserk. Now we've talked about how organelles do move within the cell, but we still want to have our rough endoplasmic reticulum and our endoplasmic reticulum next to the nucleus. We still want to have that Golgi in relationship, in proportion to the endoplasmic reticulum. And we definitely want to have the Golgi in a place where it is closer to the cell membrane than it is to the nucleus. So recap very quickly on cytoskeleton. It's very dynamic, changes in composition and shape frequently. It is not static. And it's hard for us to remember this because when we look at a cell, we are seeing a freeze frame of it when we decided to mount it on a slide and put a cover slip on it. So remember that it is very dynamic. It's not just a bag of cytoplasm within a cell membrane, okay, which is kind of a rudimentary belief or a thought maybe that we gave to kids when they were younger because it was just easier to think that way. It is a very complex system where organelles are designed, held. Yes, it changes. We have tracks via the microtubules that vesicles can move on because they have motor proteins, and it allows for movement of cell. In addition to that, some of those microtubules, again, can help to possibly act as little antennae to receive cell signals from the external environment. Okay, so actually cytoskeleton, although a lot of times very ignored, is incredibly important and really cool. So next we will do cell wall, and that will be for our next flip. Again, I told you, it's going to be a short one. Yeah.